Welcome to our research presentation on the effects of ocean acidification on pteropod density in the Indian Ocean.
I'm Bianca Broselli.
I'm Jayden Heck.
I'm Megyn Kuchinski.
And I'm Matteo, Monaca.
Let's begin by discussing ocean acidification. One of the major challenges our Earth faces today is ocean acidification, for one reason being that most of the carbon dioxide in our atmosphere is absorbed by the ocean because it is our most available sink. In response to ocean acidification, levels of pH and total alkalinity decrease. Alkalinity decreases because it is the ocean's ability to neutralize acids and bases, while pH decreases because when carbon dioxide is absorbed by the water, carbonic acid is released. When pH decreases, the amount of available carbonate ions also decreases, making it difficult for calcifying organisms to form their shell. It is especially difficult for pteropods because they use the more soluble, unstable crystalline form of calcium carbonate called aragonite. Pteropods are important to study because they are an indicator species, they are major contributors to biogeochemical cycling, and they play a large role in the food web. Studies on pteropods have mainly been focused in the polar regions because these areas are expected to be the first to experience negative ramifications due to ocean acidification. Our hypothesis is, as pH and total alkalinity continue to decrease, the impacts of ocean acidification will negatively affect the density of pteropods in the Indian Ocean.
Samples were collected via Niskin bottles from the surface waters and transferred into glass containers through a pre-treated silicone tube. These containers were then sealed to prevent gas exchange with the atmosphere. Samples were analyzed using open and closed cell titration as well as SOMA system technique. Pteropods were collected using Bagarov RAS and Jude nets within the first 150 meters and standardized to value per water volume. From our two data sets, we extrapolated our data from 3 separate years for the months of January through April. From our data sets, we were able to calculate the pH from the total alkalinity and dissolved inorganic carbon.
Our results show that from the 1970s to the 1980s, there was a significant decrease in pH from 8.17 to 8.07, as seen in figure 1. In the second figure, alkalinity significantly increases from 2,292 to 2,316 microequivalents per kilogram over the years. Figure 3 shows the significant increase in density from 0.25 to 0.52 values per meter cubed. This increase in alkalinity suggests an increased ability to buffer acidity for the increasing pH, which allows pteropod density to increase.
From our results, we can reject the null hypothesis. Interestingly, despite there being an observed decrease in pH, there was an increase in alkalinity and pteropod density. It is possible that pteropod density rose due to the increased alkalinity acting as a buffer to the decreasing pH. In other research, we found that waters with a pH lower than 8.05 negatively affected pteropod density through a downregulation of genes and shell dissolution. Decreasing pH could still negatively affect pteropod density. However, the Indian Ocean may not yet be at a low enough pH to see these effects during this time period. Other factors that could have affected pteropod density are the availability of resources, predator density in the area, and climate change, specifically increasing temperatures. Future research should look further into temperature effects, as one source found that the combination of higher temperatures with decreasing pH had a greater effect. Other future research should take into consideration other factors that have positively and negatively affected pteropod density.
We would like to thank the Biological and Chemical Oceanography Data Management Office and NOAA for supplying us with their datasets.